Module 14, the respiratory system. External respiration. When I introduced respiration to you in an earlier section, I mentioned that it occurred in 3 steps: external respiration, transport, and internal respiration. Remember, the definition of external respiration is the process of oxygen and carbon dioxide exchange between the alveoli and the blood. When blood leaves the lungs, it is nearly 100% oxygenated. Here you can see a cross-section of an alveolus. These pink ovals are cross-sections of 3 nearby capillaries. The purplish cell inside the alveolus is a macrophage. It's there to engulf any foreign invaders that might make it to the alveolus when you inhale. We call these macrophages dust cells because they utilize phagocytosis to attack small particulate that may get past the mucous membranes. Air comes into the lumen of the alveolus so that gas exchange can occur across the respiratory membrane. The alveolar fluid, with its surfactant, lines the simple squamous epithelium of the alveolus. Gases in the air move through this thin membrane and out of the alveolus. The next layer of material is the basement membrane of the alveolar epithelium. Then there's a small interstitial space between the alveolus and the capillary. The outside of the capillary has the basement membrane of the capillary endothelium, and then the simple squamous endothelium of the capillary. When you learned about the cardiovascular system, we talked about how the cells of the capillary walls are spaced a bit apart from one another, connected together by a thin membrane. Finally, the gas can reach the capillary's lumen. Now, as air enters the alveolus, oxygen diffuses across the respiratory membrane and moves to the erythrocytes in the capillary. Simultaneously, any carbon dioxide gas that's in the blood moves across the respiratory membrane and into the lumen of the alveolus. As you exhale then, the alveolus collapses a bit, and that carbon dioxide moves out of the lungs through the mouth or nasal cavity. When you inhale, the alveolus reinflates and more air enters, carrying with it a fresh supply of oxygen. No matter how physically in shape you are, your blood will always be almost 100% oxygenated. This is due to an efficient body design: 4 anatomical features and 2 physiological processes. The first anatomical feature is that the respiratory membrane is extremely thin. Thus, molecules can more easily diffuse across it. The tissue of the rest of the bronchial pathway is not made up of simple squamous epithelium. Rather, it's stratified squamous epithelium, which is much thicker. Another reason the blood is easily oxygenated is due to the large surface area of exchange. Remember, the alveoli are microscopic. There are thousands and thousands of them. That provides about 875 square feet of surface area, which is the size of a small apartment. When I talked about a person having emphysema, I mentioned that the alveoli can connect together as their tissue breaks down. Well, that reduces the available surface area, so folks with emphysema not only have difficulty exhaling due to the loss of the lungs' elasticity, they also have less efficiency in getting oxygen into their lungs. Believe it or not, though, we have enough surface area for oxygen diffusion that a person could live with almost 3/4 of their lungs gone, although they would not be able to exert themselves very much. A third reason that we have efficient oxygen diffusion is due to the capillaries being very narrow within the lung areas. That means the erythrocytes move in single file, and they're right next to the endothelium of the capillaries. Thus, as oxygen moves into the capillaries, it instantly finds a red blood cell to carry it. The fourth anatomical feature to increase gas exchange efficiency is due to the shape of the erythrocytes themselves. That little dimple inside them creates more surface area so the oxygen can get more quickly into them. Now, there are 2 physiological processes that also increase this efficiency. The first is due to a controlled connection between ventilation and capillary blood flow in the lungs. When you exercise, for example, your heart beats faster and harder. That makes the blood flow through the vessels more quickly. Well, those changes send signals to the medulla oblongata of the brain to cause your breathing to increase. So, as more blood moves through the capillaries of the lungs, more oxygen is present due to rapid breathing to supply that blood. This coordinated situation is completely controlled by reflexes. Well, what if something blocks the air passageways? If you aspirate an object, that means you take in an object by means of suction. It gets sucked down into the respiratory system as you inhale. We say a person who has aspirated an object is choking. If the airway is blocked, then no air can get to the alveoli, but the heart is still pumping the same amount of blood. The heart may even beat faster as the person realizes that he or she is in distress. But the blood can't get a good amount of oxygen because the alveoli aren't getting enough air. One easy way to remove an aspirated object is to utilize the Heimlich maneuver. This is a technique where rapid pressure is put on the abdomen of the person choking to propel the object out of the way. It's a method that is learned in any first aid class and is a potentially life-saving maneuver. This controlled relationship between ventilation and blood flow can also be messed up if a person has pneumonia. That's a condition where there is an infection in the lungs. These infections can be caused by bacteria, viruses, or even protozoans, and result in mucousy fluid filling up the alveoli, preventing good oxygen exchange in the lungs. Thus, the blood flow to the lungs is greater, and the blood is not oxygenated enough. Thankfully, with treatment of antibiotics, a person can heal from this condition. Another case of disruption of the controlled relationship can occur when the blood flow is blocked. An embolus can form if a thrombus or blood clot occurs in the circulatory system and makes its way to the heart and then to the lung. As it moves through the smaller and smaller vessels traveling to the lung, it can clog up the capillaries, reducing blood flow to the lungs. So this messes up the controlled relationship between ventilation and blood flow from the blood flow side, preventing the body from getting enough oxygen due to poorer blood flow. Well, there's a second physiological process to help in the efficiency of external respiration. It's the functional residual capacity of the alveoli. You see, even after you exhale, there's still air in your lungs. If you're sitting down right now and gently breathing, wait until you exhale and then try to force out a bit more air. You likely can do it. Most adult lungs have about 2 liters of air still in them after a normal exhale. When an adult inhales then, he or she takes in about half a liter of air at a time. We call this the tidal volume. Tidal volume is the volume of air that is inspired or expired during normal, quiet breathing. So at rest, you have 2 to 3 liters of air in your lungs at any given time. That volume changes by only about half a liter, or the tidal volume, kind of like the ocean tide moving in and out. Because the tidal volume is a fraction of the total volume of gas in your lungs, then there's still lots of air in the alveoli even after you've exhaled. Thus, during an exhale, the blood passing through the lungs can still take in oxygen. This is called functional residual capacity, or the volume of air left in the lungs after a normal expiration. This means that even after an exhale, there's plenty of air in the lungs for a good amount of oxygen transfer, and the blood can be fully oxygenated. Now, all this was when your body was at rest. What about when you're exerting yourself? During exercise, for example, most adults can take in up to 3 additional liters of air when they forcefully inhale. That means that air volume in the lungs can change from 2 to 3 liters of air to 5 to 6 liters. There's a maximum volume of air that both lungs can hold after a forceful inspiration, and it's called the total lung capacity. And just like you can inhale more than normal amounts of air as compared to when you are resting, you can exhale more than normal amounts of air too. When you forcefully exhale, you're removing as much air from your lungs as possible. Now, you can't completely empty them. There's always some there, and that's called the residual volume. Residual volume is the volume of air left in the lungs after a forceful expiration. On average, the residual volume for adults is about a liter. You may have experienced a bit more than that if you've ever been hit forcefully in the abdomen while playing ball. If the ball were to hit you there, that strong impact could cause you to unwillingly, forcefully exhale, and you get the wind knocked out of you. That just means your lungs have reached their residual volume, or even a bit below that, and it takes you a few minutes to recover by slowly inhaling and exhaling to refill them to the functional residual capacity. Now, during most of this section, I've only discussed oxygen moving into the capillaries from the alveoli. Remember that carbon dioxide travels the opposite way simultaneously. But we don't have to separately cover carbon dioxide transport because all 4 of the anatomical features and both of the physiological features that make external respiration efficient also work for carbon dioxide exhalation. Module 14, the respiratory system. Experiment 14.1, the histology of the lung. We're now going to take a closer look at an alveolus, a capillary, and a respiratory membrane by observing a cross-section of a human lung. So let's go ahead and take this slide and put it under the microscope at 40 power magnification. And what you're going to be looking for are sections of holes. You want to see white holes that are surrounded by very defined tissue. Hopefully you can see two different kinds of tissue surrounding these holes. The goal is to be able to find two holes within the same field of view that have those two different kinds of tissue. So I'm going to scan on mine until I can see that and make sure it's nice and focused. Now what you're looking at is an alveolus and a capillary. They're very different from one another based on the material that surrounds those openings, but that sharply defines them from the tissue that surrounds them. So the larger hole that you see will be the alveolus, and it's got kind of a convoluted number of cells around the inside, and the smaller hole is the capillary. So we're at 40 power magnification. Go ahead and make an illustration of what you see in your lab notebook and identify both the alveolus and the capillary. And also pay attention to the detail of the shape of those cells surrounding those holes. Now go ahead and center those ovals in the center of your field of view and increase the magnification to 100 power. So I'm going to do that right now. And focus. And we want to look at the tissue in between those as well as the tissue of those two holes surrounding them. The features of the tissue surrounding the ovals right now, let's focus on that first. The capillary is surrounded by endothelium, and the alveolus is surrounded by its epithelium. So at this magnification of 100 power, make another illustration and identify the endothelium and the epithelium that we just talked about. And then we're going to take those ovals, set them in the center, and put them at the highest power. We're now at 400 power magnification. And focus and center that in between those two. You may have to move back and forth to see all the different layers. But the goal is to go from the edge of the alveolus all the way to the edge of the capillary and figuring out the layers of the respiratory muscles or the membrane that you see as you go. You're not going to be able to see the alveolar fluid. There's no fluid visible here, but you should be able to see all of the other layers as you go from the alveolus to the capillary. Once you've completed that, go ahead and clean everything up and put it away.